Good morning everyone. Myself Sk.Ismaiel Basha, Department of Botany, K.B.N College. Today discuss about chapter 5 plant breeding. Plant breeding is the purposeful the manipulation of a qualities in plant to create a new variety with a set of a desired characteristics. Plant with higher qualities are selected by the and crossed to the obtained plants with desired quality. This result in the plant population with improved and desired traits. Plant breeding objectives. Following the major objectives of the plant breeding to increase the crop yield, to arise plants with the desired characteristics, to develop a disease resistant crop, to develop a plant that can tolerate extreme environmental stress. The plant introduction. The term plant introduction describes the movement of the agriculture plant from their original growing sites to other location. In other words, it refers to the process of the establishing crop plants in new environments. Agroclimatic conditions vary depending on where the plant variety or material is obtained and where it is introduced. A traditional technique for a crop enhancement in plant introduction. Many crops including the beans, maize, peanuts, tomatoes, potatoes, tobacco, etc. While many more fruits, vegetables, fodder crops, barley, wheat, oats, sugarcane, rice, bananas and coffee were imported to the New World. Mass selection. Mass selection is the process in which the individuals are selected from a mixed population based on the phenotype. The next generation is grown by the bulking the selected seeds. After 5 to 6 years of continuous selection, the selected seeds are multiplied and distributed. Mass selection is the oldest method of the crop improvement. It can be two types, that are a positive mass selection and negative mass selection. Next one, pure line selection. The Johansen proposed this the self-pollinated plants. The progeny of a single self-pollinated homologous plant is known as a pure line. The plant with desirable characters selected in the pure lines is called the pure line selection. He conducted selection on the princess variety of a bean, Phaseolus vulgaris. He selected 19 plants that represent the extreme seed weight from the lightest of a heaviest. When the observe the sixth generation regarding the seed's weight is the progeny, that show the same weight as their parents. The variations observed in some seeds was due to the environment. In this method, 200 to 300 plants are selected and grown separately. The superior plants are each row are identified and seeds are collected and sown in the same field. By repeating the process for the 7 to 8 years, a new variety is produced having the desirable superior characters. The clonal selection. The progeny obtained from the plant due to the vegetative is called a clone. Plants with the desirable character reproduction, a crop improvement in the clone is called clonal selection. The ginger, turmeric, sugarcane is the sets. Potato is on eye, onion is a bulb. Rose plus grapes is a stem cutting. The mango and orange is a graft cutting. The above the plant propagated by the vegetative method indicated the method adjust to them. The plants present in the clone are phenotypically and genotypically similar. Generally, there are heterozygous and shows hybrid vigor and transfer the same character throughout the breeding. The selection is always between the clones and never with a clone because all individuals in the clone have the same genotype. Hybrid vigor. The hybrid vigor, also called as a heterosis, is a term to describe the superiority of the hybrid with respect to its growth, yield and fertility over its parents. The maintenance of the hybrid vigor. The characteristics of the hybrids are best maintained by the vegetative propagation of the hybrids, as this is an asexual mode of the reproduction where the progeny contains the same genetic composition as the parent hybrid. The gene pool of the hybrids therefore remains the same. The molecular breeding. The molecular breeding may be defined as a broad sense at the use of a genetic manipulation performed at the DNA molecular levels to improve characteristics of interest in plants and animals, including the genetic engineering or gene manipulation, the molecular marker assisted selection, the genomic selection, etc. Is the molecular breeding diagram. RAPD, random amplified polymorphic DNA. RAPD markers is one of the most popular type of markers in mapping of plants genomic is RAPD. The method is the amplification of DNA of the object during the PCR with the random primer, which has a length of the 10 to 11 nucleotides. Methods of detection of RAPD markers is the easy by technique the execute. RAPDC are a DNA fragments amplified by the PCR using short synthetic primers of a random sequence. These oligonucleotides serve as a both forward and reserve primer and are usually able to amplify fragments from the 1 to 10 genomic sites simultaneously. Random amplified polymorphic DNA or RAPD markers analyze the utilize a short PCR primers consist of a random sequence, usually in the size range of 8 to 15 nucleotides in length. Complex pattern of PCR products are generated as these random sequence primers annual to various regions in the organism genome. As an extension to the variety of existing techniques using the polymorphic DNA markers, the random amplified polymorphic DNA technique may be used as a molecular ecologist to determine the taxonomic identity, access the kinship relationship, analyze the mixed genome samples, and create the specific probes. The RFLP, restriction fragment length polymorphism. An RFLP is obtained when the same restriction enzyme produces the fragment of different length from the homologous region proper of the genome of different individual strains or species. An RFLP is detected by the combination of the restriction enzyme and the probe. A probe is a short labeled usually by radioactivity DNA or RNA sequence used to detect the presence in the DNA sample of the sequence complementary of itself. The technique for the detecting and RFLP in a very brief, simple and general terms as follows. Genomic DNAs are isolated from the different strains or species. All the DNAs are digested with the same selective restriction enzyme. The DNA digested are subject to gel electrophoresis. The DNA in the gel is denaturated and transformed to the suitable solid support and fixed. The appropriate probe is used for the hybridization.